Microbiologists collect a small sample of microbes from a given source. We call a small sample an inoculum, and place it into a medium that contains the nutrients needed for life. This creates a cell culture. Culture media, also called medium, refers to a nutrient material prepared for the growth of microorganisms in a lab. Cultural media can be classified based on several parameters: the chemical constituents from which they are made, their physical nature, and their function. The medium can be in a liquid form or in a somewhat solid gelatinous form, called agar. Liquid medium is termed as broth, milk, and infusion. Semi-solid medium is somehow between liquid and solid medium on physical state, containing about 0.3 to 0.5% of agar, while liquefiable solid medium containing 1.5 to 2% agar or 5 to 15% gelatin. Non-liquefiable solid medium includes materials such as rice grain, cooked meat media, and egg or serum media. Depending on the different chemical composition, media can be classified into chemically defined medium and complex medium. The former is also named as synthetic medium, as it is prepared by adding precise amount of pure inorganic or organic chemicals to distilled water. The latter, also known as natural medium, is made from digest of microbial, animal, or plant products, such as casein from milk protein, beef extract, yeast extract, or any of a number of other highly nutritious substances. General-purpose media that supports the growth of a broad range of microorganisms. This is usually non-synthetic and often what we are using in the lab, such as LB or nutrient agar. However, some fastidious microbes need enriched media that contains complex organic substances such as blood, serum, hemoglobin, or special growth factors. For instance, the sheep's blood agar plate. Blood agar are used to determine hemolysis. The left figure points out a colony with zone of beta hemolysis when the outside circle of the colony is clear and clean. Chocolate agar is ideal for the culture of most fastidious microbial species. The brown color is the result of heating red blood cells and lysing them before adding them to the medium. It is called chocolate agar because of its chocolate brown color. A selective medium contains ingredients to inhibit the growth of certain microbes in a mixture while allowing the growth of desired microbes. For instance, gram-negative bacteria will grow on medium containing biosalts or dyes such as basic fuctions and crystal violet. However, the growth of gram-positive bacteria is inhibited. Differential media are media that distinguish among different groups of microbes and even permit tentative identification of microorganisms based on their biological characteristics. Colonies grow on the general purpose all look similar, while the same inoculum growing on differential medium exhibit different colonies. The differential medium assists microbiologists in identifying and microbial species which may be present in a sample. Eosin methylene blue agar is a differential medium. Here it was utilized to identify E. coli. E. coli is a lactose fermenter and its colonies will show green metallic sheen on the plate. Pseudomonas aeruginosa here was used as control. Its colonies were more like transparent, as it cannot utilize lactose of the media. Here are other two examples of differential media. The left figure is a blood agar. Bacteria could lyse the red blood cell and causing the clear areas around the colonies, which process is called beta hemolysis. We have already mentioned a few slides before. The right figure showed a mannitol salt agar that can differentiate bacteria that can ferment mannitol and those that cannot. Staphylococcus aureus is capable of fermenting the mannitol in the medium to acid, causing the medium to change color to yellow. Besides, it can be used as a medium to differentiate Staphylococcus and most other bacteria that cannot endure high salt concentration. As a matter of fact, mannitol salt agar can also be used to selectively grow Staphylococcus species. Another famous medium is called MacConkey agar. It could differentiate between lactose fermenting bacteria and non-lactose fermenting ones. Bacteria ferment lactose will generate pink colonies on the plate. Otherwise, the colonies are off-white. This table summarizes 6 different types of culture media depending on different purpose. A chemically defined medium is one whose extract chemical composition is known and usually used to grow autotrophs, while complex medium are used to the growth of most chemoheterotrophic organisms. Reducing medium is used when cultures of anaerobic are necessary because it contains compounds that remove free oxygen radicals. Enrichment medium is similar to selective medium but designed to increase numbers of desired microbes to detectable levels.